Examples of the impact of cell signaling can be found in every form of life on the planet, from the simplest of bacteria to the most complex of multicellular organisms. In bacteria, since you are looking at unicellular organisms that are working together, communication can typically be more about concentration of population or population density than working as a multicellular structure. Cells of many bacterial species secrete small molecules that can be detected by other bacterial cells, and the concentration of these signaling molecules enables bacteria to sense the local density, a phenomenon called quorum sensing. In response to a signal, bacterial cells come together to form things like biofilms, aggregations of bacteria that contain regions of specialized function, or to initiate some form of response. For the example that we see in the picture, we have the bioluminescence response of luciferase, which is a very common enzyme in aquatic organisms. And so the bioluminescence is only going to occur when the population density is high enough. This process depends upon what's called autoinducers, which are going to be signaling molecules whose production and proportion is directly connected to cellular density, which means that we're looking at a positive feedback loop. This can apply to a variety of other conditions, but it's really an individual versus a group behavior, and it's one way that bacteria are able to recognize the same species and others. This can also apply to things like disease. So if you have a very low bacterial count, you may not have any symptoms of the disease, but the higher the density, the more likely symptoms are to establish themselves. So through this mechanism, the entire bacterial population can respond in sync to an external stimuli, even though they are single-celled organisms. When you move on to things like plants, there's going to be a variety of responses, and researchers on how plants grow towards the light allowed us to discover plant hormones. You can see in the example here that they knew that plants bent towards the light, but they didn't know why they did it or how they did it. So when they removed the tip of the coledophile, or the plant leaf that you see here, the thing did not bend. And if the tip was covered by an opaque cap, it did not bend. But if it was covered by a transparent cap, it did bend. And if the base was covered, it did bend. So this is coming out of the Darwin and Darwin experiment from 1880, showing that what's called apical dominance, that the tip is what is controlling this process. They didn't really know what was making it happen, but the Darwins concluded again that the tip of the grass was somehow able to sense light, and the differential growth response occurred some distance below the tip, saying that the signal was being triggered or sent downwards. Later on, Boyson and Jensen tried this again, but separating the tip by a permeable substance and an impermeable substance. So with gelatin, which is permeable, the process was able to occur. But with mica, which is impermeable, the substance was unable to occur. So they went, and from this process, they identified that there must be some sort of mobile chemical substance that was producing this process. It wasn't until 1926 that they went and actually released and extracted this process, and we can see eventually this led to the discovery of the first plant hormone, which is auxin. Auxin controls the phototropic response, or that light bending. Auxins are compounds that positively influence cell enlargement, bud formation, root initiation. They encourage the production of other hormones, and they control things like the growth of stems and roots and fruits, and convert stems into flowers. How does it actually work, though? We know now that auxin triggers proton pumps. Those protons are then going to flow into the cell wall, which will increase the acidity. It's going to lower the pH, which is going to turn on certain proteins called expansins. Those expansins will loosen the connections between the polysaccharides, allowing the expansion of the cell and allowing that process to occur. So what's really happening is that the shaded side of the plant is the cells are expanding, and the illuminated side, the cells are not expanding, so it's going to lead to that growth. Other hormones do exist within plants. Auxins are just one of several. Gibberellins, for example, is one that controls seed sprouting formation. Gibberellin, we know now, for example, that the embryo of a seed is a rich source of gibberellin. So when water is absorbed, Gibberellin will be released. It will release an enzyme called amylase, which will break down the amylose. The sugars will then be used to form the initial cotyledon or seed leaves. We also see that it leads to structures like bolting on the left, where the structures are going to lead to rapid formation of floral stalks. And we also will utilize gibberellin to alter the growth of things like grapes and a variety of structures. Because gibberellin promotes the transition between vegetative and reproductive growth and are also required for pollen function, we utilize it for a variety of processes in industry. Ethylene is one that is released when plants are ripening or leading to the things of apoptosis or the loss of leaves in fall, for example. It's going to apoptosize from the leaf separating it. But the ethylene is a gas, and the gas is an active factor leading to a variety of responses. It instigates seedlings to perform a growth maneuver called the triple response that allows seedlings to circumvent obstacles such as stones that might grow in their way. The ethylene is going to slow the elongation of stems, it's going to thicken stems, and it's going to alter curvature, allowing it to start growing horizontally. So plants will utilize ethylene growth to get around some sort of barrier. But ethylene is also used for things like fruit ripening. Enzymatic breakdown of cell wall components soften fruits and the conversions of starches into sugars, making fruits sweeter. So we will utilize ethylene balance to prevent fruit ripening in transit. But then we will then encourage ethylene when vegetables get to a store, for example, so they ripen for sale. We see other responses exist outside of the hormone responses. Plants, for example, have an immune response called a hypersensitive response, where pathogens will enter cells and they will then be recognized by what's called the R proteins. The R proteins are going to then initiate a signal transduction pathway that will alert the plant to this invader. This HR, or hypersensitive response, will be initiated with that binding to R protein, and it is going to stimulate the production of things like phytoalexins, which are toxic compounds with fungicidal and bacterial properties, but will also increase the production of lignin and cross-linking molecules, essentially walling off infected areas, as you can see on the right, and killing off the cells that are inside the walled pathway. This will also send signal throughout the rest of the plant and into the roots, connecting to other plants, allowing for the systemic acquired resistance, the long-term process, which is going to lead to the production of things like salicylic acid in remote sites, so that when the pathogen gets there, defense mechanisms are already in place. In animal cells, the most important series, arguably in early processes, is looking at stem cells and the overall determination and development of the embryo. If you don't know, in mammals, embryonic cells remain totipotent through the 8-cell stage, much longer than many other animals, but the totipotency is not fully equivalent in a normal embryo. Totipotent cells can become any structure within the organism or the developing process, but as cells undergo division and differentiation, they become increasingly less totipotent. You might become oligopotent, multipotent, pluripotent, okay, the sort of various levels of response. But during development, cells become specialized, undergoing differentiation, which will then organize into tissues and organs. The physical processes that will then alter its shape are called morphogenesis, creation of form, and that's going to lead to embryonic development. Can we reverse this process? Artificially, yes. You can take a unipotent cell like a skin cell or something and turn it into an induced pluripotent stem, but those cells are going to be artificial within process, so they don't tend to be as effective as true stem cells. And we do have some control mechanisms where the induced pluripotent stem can fail, but it is sort of our best attempt. Some genes are positional in nature. These so-called egg polarity genes are morphogenic in change, and the best example that we have of this is the bicoid gene in fruit flies. Bicoid is a positional informer that's encoded by the embryo's genes and establishes a specific number of correctly oriented segments. This process determines the anterior-posterior relationship, so basically wherever the bicoid is concentrated will become the anterior end. If that gradient does not exist, then the developing egg will typically fail. So morphogens are a great example of what we call cytoplasmic determinants. These are going to be molecules that will determine what the cells of the cytoplasm will become. So cytoplasmic determinants are important sources of early information in the development of the egg cytoplasm. They contain both RNA and proteins encoded by the mother's DNA, and they are typically distributed unevenly in the unfertilized egg. Once the egg actually fertilizes, the cell will begin to divide, and that differentiation will set the initial process of determining what the cells will become. These signals will include contact with cell surface molecules on neighboring cells and the binding of growth factors, which will then lead to changes in nearby cells in a process called induction. The molecules conveying these signals within the target cells are then going to be expressed by the embryo's own genes and sending cells down specific developmental paths. Basically, cytoplasmic determinants are specialized and they play a very important role in the oocyte maturation, and this process is going to lead to the original or the basis of the positional information and pattern formation of the embryo. This will then overall lead to changes in the structure and function of cells, the morphogenesis, or the movement and formation, leading to the actual development process, which we'll look at next. Because almost all cells in an organism have the same genome, differential gene expression results from differential gene regulation in different cell types. And during embryonic development, cells become visibly different in structure and function. Molecular changes in the embryo, called determination, lead to observable differentiation of a cell. Once it has undergone determination, an embryonic cell is irreversibly committed to its final fate using natural means. We can do it artificially, but otherwise the cell is determined. The outcome of determination, which will be observable cell differentiation, is caused by the expression of genes that encode tissue-specific proteins. So determination in this example, the embryonic precursor cell becomes determined to become a muscle formation by the activation of regulatory genes that will produce things like myosin or myo. That determined cell is now a myoblast. It is a determined cell that is less potent than it was before. It's moved down its level. So instead of becoming any type of cell, it can now become any type of muscle cell. The master regulatory genes will then activate a cascade of lower level specific genes, which then turn on and allow for the production of specific proteins like actin, myosin, and all these various structures. So determination, then differentiation. From differentiation will lead to physical changes, morphogenesis, and development of the embryo into you. In some cases, you actually have the opposite situation. Some cells are programmed to die. Programmed cell death, or apoptosis, is a common feature of animal development, and during development, individual cells or sets of cells, or in some cases whole tissues, will cease to develop and are engulfed by neighboring cells. One familiar example is the tail of a tadpole, or the separation of your fingers, for example. The interdigital tissue will be apoptosized away, leading to the formation of the individual structures. Now, apoptosis is a programmed process. It is steady. The changes are including what's called blebbing and cell shrinkage and nuclear fragmentation, chromatin condension. It's a very unified process. This is different from necrosis, which is essentially a death and a random breakdown, which you can see differently in the picture on the left. Apoptosis is extremely important in vertebrate development and maintenance, with several different pathways involving about 15 different proteins carrying out this process. These signal processes are incredibly elaborate and can be very complicated to observe, but the life or death question is the most fundamental one imaginable, really, for the cell itself. An animal hormone is a chemical signal that is secreted into the extracellular fluid, circulates in the blood or hemolymph, and communicates regulatory messages. Chemical signals by hormones is the function of the endocrine system, one of two basic systems for communication and regulation throughout the body. The other one is the nervous system, which is going to be a network of specialized cells called neurons that will transmit signals along dedicated pathways. These structures will work together to regulate body structures, with the example here coming from the brain being a neurosecretory cell from the hypothalamus releasing a control hormone, or in this case TRH, or the thyroid releasing hormone. That will then travel through the bloodstream to the thyroid. The thyroid will then be activated through this thyroid stimulating hormone coming out of the pituitary gland to secrete hormones like T3 and T4 to initiate metabolic responses. So if you have a thyroid issue, it could be any one of those steps that fail you. But overall, we find that the hypothalamus is one of the major regulators, and the pituitary glands at the base of the hypothalamus are then going to control the release of stimulating hormones. So you have the releasing hormones and the stimulating hormones working together. An example is ADH, or the antidiuretic hormone. When your blood osmolarity increases, so you've lost a lot of water from, say, sweating profusely, the hypothalamus will say, hey, posterior pituitary, release ADH. The ADH will then alter the kidneys and generate that desire for thirst, which is going to cause you to absorb more water and drink more water, bringing your blood balance back to normal. So hormones are going to be controlling a variety of physical and chemical responses throughout the body by traveling through the bloodstream or through bodily fluids or tissues. Another example is the digestive system. So when food enters your stomach, it's going to cause a positive feedback loop of a hormone called gastrin. Gastrin is then going to stimulate the release of things like hydrochloric acid and pepsin to digest materials in the stomach. But once that food leaves the stomach and enters the duodenum, the early part of the intestines, you then are going to be looking at the digestion process in the intestines. That's going to be triggering the liver to release bile stored in the gallbladder. That is going to help break down fats. Okay, CCK is the hormone that triggers that release. You also have secretin and CCK, which will activate the pancreas to release bicarbonate, which will neutralize the acid of the chyme from the stomach acid, and various digestive enzymes to do most of that digestive work. That will also trigger the negative feedback of the gastric juice production, because once you no longer have food in your intestines, you don't want to keep releasing those acids and lead to things like ulcer formation or autodigestion. So hormones are going to be tied together into a lot of these things. Other ones are going to be released by these organs and will be controlling the brain process, with organisms like hormones like ghrelin being incredibly important. Ghrelin is produced in the stomach, and its function is to tell the brain that the body has to be fed. It increases your appetite, with gastrin being produced when the stomach is stretched. It stimulates the release of gastric juices, and secretin is going to be interconnected there. Overnourishment causes diabetes, obesity, and that kind of structure, which is going to be a breakdown of these processes. Researchers have discovered several mechanisms. Ghrelin, for example, triggers a feeling of hunger before meals. Insulin and PYY, which is secreted by the small intestine, suppresses appetite. And then you have leptin. Leptin is produced by adipose, and it suppresses appetite and may regulate body fat levels. But if you have excess fat deposits because of obesity, that leptin is going to be triggering the brain again and again and again and again, and your body will stop responding to that satiation center. So like type 2 diabetes, where you have overexposed your body to the insulin and you're no longer responding to the blood sugar sensations, leptin is going to be over-triggering, and you're not going to feel full as easily, which is going to be a positive feedback loop leading to obesity. So there's a lot of hormonal balance and impact here that we need to look at as we look at whole health rather than organismal structure. Female reproductive system is going to depend upon a variety of hormones, but they're going to be ultimately controlled by the hypothalamus. The hypothalamus releases the gonadotropin releasing hormone, which will then trigger the anterior pituitary to release FSH, or the follicle stimulating hormone, and LH, the luteinizing hormone. Those are going to control the growth of the follicle where the egg can be found, and whenever the egg is going to be released, that LH surges. Okay, so the LH surge is going to cause the release of the egg from the maturing follicle. okay, and that's going to lead to the alteration of estradiol production. You're going to have progesterone and estradiol, which is going to be released by the corpus luteum, and those will then lead to the development of the uterine lining and the uteral layer. But if the egg is not actually embedded, then those hormones will fail, and the endometrium, the uterine layer, will be shed, and the female will experience the monthly cycle, the period. Those same hormones are controlled in the male reproductive system. Again, they don't have follicles, but they do have gonadotropin-releasing hormone, FSH and LH. Instead of producing estradiol, they control different cells in the testes, the Sertoli cells, and the Leydig cells. The Sertoli cells support the germ cell progenitors and help transfer nutrients from nearby capillaries. The Sertoli cells produce testicular fluid, and they include proteins that bind to and concentrate testosterone. The Leydig cells are interstitial, they lie between the tubes, and they have this structure that is going to lead to, again, the production of testosterone, and those will work together to produce sperm. Okay, if there is an abundance of these testosterone release, the structure is going to have a negative feedback loop, which will slow their production, tying them around. But these negative feedback mechanisms maintain androgen production in males. Other hormonal connections is going to be like the clotting formation. So whenever you have a wound, platelets and damaged cells are going to release clotting factors. Those clotting factors will trigger an enzymatic cascade that will turn prothrombin, which is a soluble fiber protein, into an insoluble thrombin, which will cycle and lead to clots. Okay, that is going to lead to the upregulation of fibrin, which will create those fibers that you think of in sort of a scab structure. And inherited defects in any step of these processes can lead to something like hemophilia. You also, of course, have the nervous system, which is going to be tied into sensory response. You're going to have some sort of stimulus, which will be picked up by a receptor, or maybe by some sort of neurotransmitter producer. Those are then going to send signals to the central nervous system. And if you notice the picture on the right, the signals are always going to be the same action potential spike, and a stronger stimulus will just generate more signals, not a stronger signal. So for example, the hairs in the inner ear, which we can see on the bottom right, fewer hairs being triggered will be a lighter sound, whereas a louder sound will trigger more hairs, will trigger more responses, but the action potential curve stays the same. Breathing control is another example of this, with the hypothalamus measuring the blood pH, okay, and the medulla oblongata and the cerebral spinal fluid, and as well as cells in the heart and throughout the body, basically taste receptors, are going to be constantly measuring the pH throughout. Those structures are going to be controlling your breathing rate. So whenever the pH begins to go down because CO2 turns into carbonic acid, you start breathing faster to ventilate to remove that CO2. So if you hold your breath, your brain starts getting upset because of a pH imbalance because of CO2. Immunity is another place where reception and transduction response plays a major role. Pathogens that penetrate the first line of defense are subject to phagocytosis, and phagocytic cells detect fungal or bacterial components through receptors that are called toll-like receptors. So they're going to be looking for things like flagellin, which is the protein that makes up bacterial flagella, not ours, or maybe double-stranded RNA, which is not something that we use as self. Those are then going to trigger lysosome responses that are non-specific. We also have antibody production, looking at B and T cell formation. They are going to create these variable regions of binding that are basically random in formation, and that will then lead to recognition and response. Those things take advantage of what's called MHC cells, or major histocompatibility complexes, and those are going to take pieces of antigens that are inside the cell and put them on the surface so that the T cells can recognize non-self DNA without having to kill your cells. So this reception response process, these antigen presenting cells are a major component of that recognition. So So we find overall that there are many different types of cells that are responsible for the immune system, but B and T are the most important. Helper T, for example, will work with the MHC, and the helper T will trigger the release of enzymes like cytokines. Those things can then trigger B and cytotoxic T cell responses, or both of the major forms of immunity. And this is one reason why HIV is such a problem, because the HIV virus grows inside these CD4 T cells. Since the helper T is central to all of the other immune responses, your immune system begins to fail. Helper T cells will trigger the B cells to release antibodies. Those antibodies will then bind and lead to the digestion of invading bacteria, but will also be stored in memory B cells in your lymph nodes, whereas the cytotoxic T cells are going to be releasing enzymes like perforin and granzymes, which are literally going to poke holes and begin to digest bacteria that infect the cell. But ultimately, all of these actions are just applied examples of cell-level functions that then can affect the entire organism.